No brown spots? You got it. Want turnips that look light but feel heavy? Easy. Want Honeycrisp apples that are firm, green, and definitely not Macintosh like last year when you lost the fall bake-off to perfect Penelope Johnson? Okay. A bit TMI, but we're here for it. So download the app today and get $0 delivery fees on your first 3 orders. Instacart. Groceries that over-deliver. Service fees, exclusions, and terms apply.
I'm Flora Lichtman, and you're listening to Science Friday. Today in the pod, big news in the food allergy world.
This gives us a little bit of hope that we are making a dent in food allergy.
Some welcome news. A study in the journal Pediatrics found that peanut allergies in babies and little kids are on the decline. And this drop correlates with a guidance change from the National Institute of Allergy and Infectious Diseases. In 2017, they started recommending that we expose children to peanuts early and often. And since that recommendation, the prevalence of peanut allergies has dropped significantly. And that's a huge shift because for many years, peanut allergies were on the rise. Here to churn through the findings is Dr. Sharon Shinthraja, a physician specializing in allergy and immunology at the Sean N. Parker Center at Stanford University. Sharon, welcome to Science Friday.
Thank you, Flora. I'm so excited to be with you today.
So what did the study find exactly?
So what the study looked at is the rate of food allergy prevalence, how many people have food allergies, by studying the electronic medical records before the guidelines were changed, and then immediately after the guidelines were changed, and then a little bit post that period as well. And by looking at that, they found that the rates of diagnosing food allergy has dropped after the guidelines were published. So that's super exciting because the guidelines were based on NIH funded research, and that's, very solid data and evidence that if we introduce peanut early in the diet for infants, for babies, that we can actually prevent the development of peanut allergy. And that was a huge study that led to the guideline changes. And so what it says is that doctors are doing a good job, families are doing a great job following those guidelines or knowing about those guidelines, how to feed their babies, what to feed their babies to make a difference to prevent food allergies.
Tell me a little bit more about this. Where did this idea of microdosing peanuts to babies come from?
Oh, it's such a great story, Flora. This is why I always tell, my kids and students to be very curious. So Gideon Lack, who's the last senior author of the LEAP study, this study that I was talking to you about, and other researchers noticed Jewish babies in Israel didn't have food allergies, but Jewish babies in London and UK, where he was located, had food allergies, had peanut allergies. So even though they shared similar genetic backgrounds and probably cultural backgrounds, there was something different about the environment that was showing more peanut allergy in London than in Israel. And when he went to go visit colleagues and friends there, what he noticed is that families were feeding babies peanut bamba. And now, back in the day, nobody would know what peanut bamba is. But now, because of that
Every
study,
parent knows what peanut bamba is now.
Exactly. And there's even different flavors, right?
These are they're like Cheetos, but they're covered in peanut butter, basically.
You're exactly
And they're
right.
Israeli, right?
Yes. And now there's all different types of brands. And the beauty of it is that it melts in your mouth, right? And so for babies, it's not a choking hazard, right? So that's the key piece there. And so babies were holding on to these little puffs, and it was dissolving in their mouth, and they were getting good oral and gut exposure to peanut proteins early in life. And that is so important. Turns out that exposure early in life, in that critical window when you start to introduce foods into the diet, is so important, because guess what? 70% of your immune system lines your gastrointestinal tract, lines your gut. And so you are training your gut that peanut is fine, peanut is okay, peanut is harmless. Introducing peanut as early as 4 months of age and keeping it in the diet, it reduced the risk of developing food allergies at the age of 5 by 80%.
Wow, 80%. That's amazing.
Huge.
Yeah, huge.
Just by this simple change in dietary introduction.
Can I ask you a question? Why are peanuts such a trigger for the immune system? Like, what is it about the humble peanut?
Oh my gosh, this is $1 million question, right? You know, the immune system is tricky. It's meant to be against harmful things in the environment. And so different than the types of antibodies we use to fight off bacteria and infections. And it's an age-old system really meant to protect us against parasites. And the IgE antibody recognizes proteins in the environment. So if you have seasonal allergies, it's recognizing proteins in pollens. So if you have food allergies, it's recognizing proteins in food. And the reason, many reasons, you know, I think we're becoming more allergic as a society, and there's environmental influences and genetic influences there. But the way the body and the immune system sees the food is important. And it turns out that if you have peanut in your household, mom, dad, sister, grandma, that you can find peanut protein in the dist in your household.
Is it just very durable? Is that the idea? Like it holds up in the environment?
It holds up, and guess what? So that dust can be exposed to your skin. And if you have eczema, a different type of allergic disorder, your skin barrier is not perfect. It's leaky. And so if your immune system sees peanut protein, you know, more commonly through kind of this leaky skin, it actually trains your immune system to develop an allergy. So it's even more important that we train it via the gut.
So your immune system is looking for these proteins. I mean, is that why eggs are also problematic because they're protein rich?
Yes. So not all food proteins are equal. Egg, I think, is probably one of the first foods that are introduced and very common in the household because of baked products. Eggs is a good source of protein. Milk is another one that is common early in life. You know, but what's really interesting is that in the over 10 years that I've been really studying food allergy, different allergens are popping up because society is more interested in enriching protein in our diet, and healthy proteins like tree nuts and peanuts, which is actually a legume. And so we're starting to see different types of allergens early in life as triggers for food allergy.
Like almond butter or cashews or walnuts or something, because people are using these more in food.
For sure, cashew and walnut are becoming more prevalent than 10 years ago. And you know, the initial guidelines changed in 2017, and then the NIH and expert panels put together a recommendation to introduce peanut early into the diet. It changed in 2021 to liberalize that. All foods, you should have a diverse diet early in life, not just tree nuts. And that's really important. This new study published in Pediatrics, also funded by taxpayer dollars, and the NIH, shows that we blunted the rise of food allergy.
Right, it wasn't just peanuts, right? It was other foods too.
It was other foods too. And but it's still there. And so early introduction is not enough for everybody, but it is now flattening the curve. Prior to the guideline changes, it kept on rising. You know, the number of food allergy diagnoses was on the rise. And so at least we've blunted that. right? And we need to look and see, have we blunted that for all of these other foods?
Do we understand what's susceptible?
That is a great question. We know that food allergy occurs in babies and children that have already kind of this allergic immune system, and they have other allergic diseases like eczema or atopic dermatitis. And we know that babies can show signs of allergic inflammation, even in the first weeks of life, so that there's a skewing of their immune system because they have dry skin or eczematous skin. And so the question really then is, how early should we be introducing foods into the diet, and how? And we need to figure out ways how we can do the introduction of foods earlier than 6 months, earlier than 4 months, in a very safe way.
We've covered other large epidemiological studies on the show in the past, and we know how hard it is to actually sort of like have a study where we're like, okay, case closed. And I know in this study, they looked at electronic health records, not actually at what the kids ate. And we know that the timing lines up so that it suggests that it is linked to these new recommendations. But do we know for sure? Is there more work to be done?
You hit it on the nail. One of the limitations of this study, Flora, is that it looks at electronic health records. The gold standard of diagnosis is a food challenge, where we're actually introducing the food and watching for an allergic reaction, which is often done in the allergist's office to manage allergic reactions in case. But I think this gives us a little bit of hope that we are making a dent in food allergy. We're working on better diagnosis other than the food challenge, and now that there are treatment options available. So we're getting there. Food allergy is a chronic disease once you have it. We don't yet have a cure, but we have more treatment options available so that people don't have to suffer with it compared to earlier days.
Dr. Sharon Shintrada is a physician specializing in allergy and immunology at the Sean N. Parker Center at Stanford University. Sharon, thanks for joining me.
Thanks so much for having me, Flora. It was my pleasure.
Today's episode was produced by Rasha Arudi. I'm Flora. Hey, it's Flora Lichtman. You're listening to Science Friday. On today's show, zooming in on the sophisticated secret societies living around and within us.
They've been here for billions of years. They do evolution on a much faster scale. So they've had time to occupy every niche on the planet and to optimize.
As humans, I think it's tempting to imagine ourselves as the pinnacle of evolution. We talk, we make art, we build cool and transformative things, and we change the planet in profound ways. But there are life forms that have been on this planet evolving a lot longer than us, like way longer. Bacteria go back billions of years. What if they have come up with sophisticated playbooks that we just don't yet understand? Could they have their own culture, their own language, their own complex societies that we're ignorant of, even though they're playing out right under and perhaps even in our noses? Those are some of the questions my next guest has been looking into for more than 30 years. Dr. Bonnie Bassler is a microbiologist and
Thanks, Flora. I'm delighted to be here.
What did you think of that intro, Bonnie, about how much of what bacteria are doing do you think we're unaware of?
A lot of what bacteria are doing, we're unaware of. We know a lot about the parts list that make bacteria and other organisms, but the behaviors, the sophisticated, magical, astounding things bacteria can do, that's been the focus of a lot of work for the past few decades. And in that realm, I think we're only beginning to understand the complexity.
What about language? Is there an argument that bacteria have language?
Well, since that's my life's work, I would say yes. And so, of course, bacteria, right, so just to set the tone a little bit or set the stage a little bit, of course, bacteria are microscopic. They're single cells, right? You can't see them with your eyes. To see them, you need to look under a microscope, right? So they don't have language the way you and I do. They don't talk with words, but what the field has shown is that they actually communicate with chemicals. So they use chemicals as their words, and they use those chemicals to count how many neighbors are around, to take a census of who those neighbors are. Are they friend? Are they foe? Are they relatives? Are they not relatives? And then they use the information, all of which is embedded in these molecules, to help them make decisions based on whether they're alone or in groups and who's in the neighborhood.
How well do we understand how that chemical sensing works?
So we actually do understand a lot about that. So this field is a very vibrant field. The field is called quorum sensing, and that the bacteria sense when they're in a quorum, and then they do things as groups. And we understand some about what the molecules are. We know some of the molecules. We understand something about how they're detected. We understand something about how the bacteria integrate the information and how they decode the information that's embedded in these molecules. And we know a lot about what they do when they're alone and when they're in groups.
I mean, that sounds like they're sending messages, but are they also conversing and forth? Like, does the relay go more than just one stop or two stops?
Right, so it's not exactly a conversation. Like, you ask me a question and I give you a different answer, and then you interpret what I say and ask me the next question. So far as we understand now, and of course, for sure there's more to find out, what we think that they're doing, the simple things they're doing is they're using molecules that build up outside of the cells as the cells grow. And so the molecules, the amount of molecules around the bacteria track with how many bacteria are there. So first they use it to count. They say, am I alone or am I in a group? Because they want to behave differently when they're alone and when they're in groups. You know, when they're alone, they carry out tasks that a single bacterium can accomplish, but they don't bother to do tasks that it takes lots of bacteria acting together to make the task successful unless they're in a group. So the first thing they do is they say, am I alone or am I in a group? And then they do tasks A, B, or C, or D, E, or F. And those tasks become successful depending on how they get carried out. And then the second, more sophisticated thing they do with those molecules, because there's a blend of these molecular words, they ask, who is around me? Is it my twin? Is it my cousin? Or is it the enemy? And then they change the tasks that they carry out based on whether they're surrounded by, family and friends or the enemy. So they share and they do sort of nice group behaviors when they're surrounded by their siblings. And then when they're in competitive environments, they try to kill the other guy, or they try to keep their goodies to themselves, and they're not as available for sharing.
Give me an example of a behavior that they would do just with friends and family, and then a behavior they would do amongst folks.
So they need to take little bites out of it. And so what they'll do is they will secrete enzymes outside of themselves that chew up solids into small enough bits that but of course, if you put your enzyme out, it and your neighbors can get them. So they're called public goods, because they're going to make an expensive enzyme that they have to put out of themselves to chew up the solid food, and then anybody can get the products. Okay, so you want to do that if you're surrounded by your, it's like you're feeding your family, right? It's a group dinner, family style dinner.
Sunday dinner, yes, we've all been there.
Yes, exactly, right? Dig in. Okay, so that's something, for example, public goods and sharing the effort of your work. Bacteria will do when they're around their families. But you don't want to share if it's your competitor that's eating up all the food. So they typically won't do those kinds of behaviors, like public good behaviors, when they're surrounded by foe. And so in that case, they'll do other things, like make poisons that they are immune to and that kill off their competitors, right? They put them out and then they try to, outwit or get rid of their competitors. And so those are all group behaviors, right? But it matters who's in the neighborhood, which set of group behaviors get taken.
And are they sensing other bacteria? Do they recognize viruses?
Yeah.
What do they know about who's around them?
Right. And so that's actually the forefront of this field. And so if I go back a little bit, we used to, in the beginning of this field, I should just say it was astonishing. 30 some years ago, or even more than that now, when it was discovered that bacteria had the capacity for group behaviors. It was always thought that bacteria were sort of asocial and dumb. They divided in half and each bacterium did its own thing, that they didn't have the sort of genetic capacity to know there were others around. And so in the beginning of this field, the first thing scientists thought was, when they realized there were molecules that these bacteria were releasing, the molecules were increasing in proportion to cell number, and then the bacteria would do behaviors as a group. The first thing we thought is that they know that they're around their siblings. Then after that, we realized there's more than one molecule in this language. Then there was a molecule that was for the cousins. So first there was the species, then there was the genera, the family, and then there was an interspecies molecule, so they could talk across species boundaries. And now what we've learned, and this is only in the last few years, is that they can actually detect if they're in a host, so they can detect molecules made by bacteria, just like we are bombarded by viruses. Bacteria are also attacked by viruses all the time. And so in fact, evolve the capacity to listen to these bacterial conversations. They're eavesdroppers. They can recognize when there's lots of bacteria around, that's a good time for the virus, the predator, to attack the bacteria. So now we're thinking that these quorum sensing conversations span all domains, from viruses to bacteria to eukaryotes, to mammalian cells, right? And so the field is still in its infancy in a way, even though we do know a lot about the molecules and how they detect them and how they, decode information. I still think, well, I need a job. And so I still think that these bacteria, they were only at the beginning of understanding the complexity and the capacity of these chemical conversations.
Well, what I'm thinking is like these bacteria inside me know more about me than I know about them.
No question. Right? But they've had a longer time, right? So they've had billions of years, right, to evolve. In the recent history, and they have been evolving in collaboration or in competition with higher organisms, right? But yeah, so they're tuned in to all of this information. They're sort of like little computer chips, right? That they're taking in all kinds of information, and then moment to moment to moment, they turn on and off genes, which lets them turn on and off behaviors, and that's how they succeed. And it's very similar to what we do. It's sort of a stripped down version of us.
Well, yeah, that's the thing. I mean, I'm not to be all big thinky here, but, I think we feel like we have a monopoly on decision making.
Yeah, we think we're these rarefied, special organisms on Earth, and I do think I'm charming.
You are special, Bonnie.
Yes, thank you very much. But anyway, yeah, so the truth is that we evolved from bacteria. Every organism evolved from bacteria. They've been here for billions of years. They do evolution on a much faster scale because they divide every 20 or 30 minutes than human beings or higher organisms do. So they've had time to occupy every niche on the planet and to optimize, right? And this sort of logical decision making that you're talking about, Flora, in a bacterium, they don't have brains, they don't have consciousness, they don't have feelings, like we do. We are special, okay, in some way, but in the simplest sense of the biochemistry and the genetics that goes on in any living organism, these decisions are very analogous to one another. Information comes in, and then bacteria and we behave, right, in a way that, in the case of the bacteria, that allows them to succeed.
We're going to take a quick break, but don't go away. When we come back, how Bonnie got into this field.
It started as simply a question about how can a bacterium be part of a group. They seem to be too primitive and too stupid to be able to do that.
I'm Samir Patel, editor-in-chief at Quanta Magazine. Our mission at Quanta is to cover what we call basic or fundamental research in science and math. That means the kind of work that's driven by curiosity and discovery and the search for knowledge. We're interested in how and why everything around us works the way that it does. On our brand new show, the Quanta Podcast, you'll meet the writers and editors behind the magazine, and together we'll explore the biggest ideas and the tiniest details. Join me for the Quanta Podcast, your weekly dispatch from the frontiers of science and math.
I mean, do you think that my anthropomorphizing questions are problematic? Like, where do you fall on that idea?
No, I anthropomorphize everything because I think it's an easy way to understand it. They are communicating. They are knowing friend from foe, family, they're making decisions. I think thinking about bacteria and their language, which we've talked about, is chemical. It's not words, it's chemical, but the chemicals are the words, and being able to extract from those chemicals not only how many neighbors are around me, but who the neighbors are. I think anthropomorphizing that is a good idea because it is the progenitor of our language and our ability to know self from other, friend from foe, and to make individual decisions and group decisions, right? And so I don't think that's a wrong way to think about it.
It's the foundation for our own behaviors.
They are the foundation for us. So yes,
Yeah.
I think, okay, I'm brainwashed. I do think it is the foundation for our own behaviors. And then when we really think about like what these bacteria are trying to accomplish, right, by deciding, can they do something alone? Can they do something in a group? We make those kinds of decisions all the time. Like the behaviors did build on one another through evolution. So why not think about them, use similar words to describe them as we do for, say, humans?
Yeah, we need to bacteriomorphize ourselves or something. Personally, how much are you driven by practical applications? Like, that cracking bacterial communication could help fight diseases.
Well, so that is a big theme of my team's work. I am a do-gooder at heart. And like if I really confess to you, when I started this, that's not what I thought. This whole field started in an obscure but beautiful bioluminescent bacterium that what it did as a group was turn on light. And that made the invisible world of the bacteria visible to the scientists. And it gave us something to track. We could see that they only made light as a group. And so my question back then was, how do they know to do that? How can they do that? This is how bacteria get any bang for their buck. They know when they're alone, they know when they're in groups, and they act accordingly. But back then, there was no biomedical or industrial or ecological significance to the work. It was just the example that we had. And what we thought back then was like, we wanted to understand, how does any collective behavior work on Earth? How can organisms carry out tasks in groups and accomplish things that they couldn't accomplish as individuals? And of course, for me, working on bacteria, they're very, not that anybody believes this after this conversation, they are much simpler organisms, right? They're fast growing, they're clones of each other. You can have, a surprise in the incubator every 8 hours to do your studies, right? And so that was just super attractive to ask questions about collective behaviors, just as what we would call a model system for group behaviors. Then lo and behold, what was discovered in these bioluminescent bacteria ended up being discovered in tens of thousands of kinds of bacteria, including pathogens, including industrially relevant bacteria. And we now know that being able to, I liked biology classes, I liked chemistry classes, and there was a bulletin board that had professors that would let you work in a lab. And so I literally went to this lab, and the professor had a bacteria project and a cancer project. And so I thought, I was 19 years old. To the important project. Well, Flora, it's a few years later, and perfect each of them for their own niche. And bacteria have perfect each of them for their own niche. And bacteria have been model systems. That's how we know about DNA, RNA, and proteins, and the molecular biology revolution. It all came from bacteria and viruses. And so the truth is they're not solved yet, right? And so it was thought sort of that the bacteria gave us the parts list, like I just said, DNA, RNA, proteins, and that higher organisms were going to give us all the cool stuff, behaviors, appendages, body parts, development. So there was sort of a point when bacteria were a little bit out of vogue. That turned to be right when I was starting my career, because it was thought that the good secrets had already bacteria possessed had already been found. But lo and behold, scientists kept at it, those of us who loved bacteria, and then we found out that beyond giving us the amazing parts list, right, of the parts that make a living organism, they do have all these behaviors, the one we're talking about, communication, collective behaviors, right? They have body plans, they put the right things in the right places, just like you have arms, legs, head, in the right place at the right time. Bacteria, it turns out, they were just so small you couldn't see that back then, so they do all of these terrible and magical things. So I think for me, it's given me, a way to study those kinds of logical questions in this simple system. That's a long answer from me. Yeah.
Before we let you go, what is your top burning bacterial question? Like, what is the thing that you want to know above all else before you leave this mortal coil?
Yeah. So they've showed that with viruses, with higher organisms, in space and time, not all interact with viruses, with higher organisms, in space and time, not all around in a perfect environment. Because if we're really going to make applications, like either turn on chit chat when we want or stop harmful bacteria from talking, it's not going to get done in a test tube, shaken around in a perfect environment in Princeton, New Jersey, right? We have to be able to learn enough about this, how this works in the real world, that we could safely and reliably and successfully manipulate it if we want to make applications. And so going back to the first thing you said, for us, it's all about complexity and trying to think of ever more complex environments or scenarios to see how could this quorum sensing and bacterial communication and group behaviors ever work outside of a test tube in a lab. It does. We know it does, right? But yeah, I think that's our next 10 years.
I can't wait to have you back to talk about it.
Well, that would be a delight, and I can't wait for that. I hope it's not 10 years.
Thanks, Bonnie. Hey, I'm Flora Lichtman, and you're listening to Science Friday. Today on the menu, the future of food, including a serving of lab-grown fish. What does it taste like?
It was bright orange. My first thought was, oh my God, this is Nemo on a plate.
If you order salmon at a restaurant, you often have options. Sockeye, coho, Atlantic, farmed, wild caught. And in 4 restaurants across the country, you may also have another option you haven't seen before. Cultivated salmon, also deliciously known as lab-grown. Food correspondent and adventurous eater Kathleen Davis, a sci-fi producer, sampled petri dish fish at a high-end sushi restaurant. She's here to tell us the fish, take us on a deep dive into the state of the lab-grown fish industry. Hey, Kathleen.
Hey, Sarah.
Okay, set the scene.
So about a month ago, I was in San Francisco, and I had quite the West Coast techie experience. So I took a self-driving taxi for the first time. I'm scared. But it's actually a very smooth ride. I feel actually like I'm in good hands. Flora, I don't know if you know this, but they're straight up piping spa music in those cars as, I think, a relaxation tool.
I would need that to help relax me.
I did. I did. But my car did deliver me in one piece. Robin, which is one of the 4 restaurants in the U.S. that is serving cultivated salmon. I've been covering lab-grown meat for a while, but mostly it's been in the context of things that are meant to be cooked. So think like a beef patty or like a piece of chicken. This was going to be raw.
Did you have any hesitation about trying it?
Honestly, no. I was just excited to be along for the ride, honestly.
Okay, so take me there.
Okay, so the chef at the restaurant, his name is Adam Tortosa, he served me up 2 pieces of sushi. So one was traditional salmon, and the other one was cultivated salmon, which is made by this company called Wild Type. So Wild Type is the only company that is making and selling lab-grown fish to restaurants in the U.S.
Okay, and how are they serving it at Robin?
So the fish at Robin is served nigiri style, which is like a strip of raw fish on top of a little rectangle of rice. So I'm sitting at the bar, Chef Adam brings out the wild type salmon, and I have to say the first thing that I noticed is that it has a blinding color.
Like blinding how?
It was bright orange.
Like in a yummy way?
My first thought was, oh my god, this is Nemo on a plate. Like, it really did kind of look like a clownfish. It was bright orange. It had those little strips of white fat running through it, which is similar to regular salmon, but that is the headspace that I was in going into this. Chef Adam did say when I commented to him about this that orange color is a little closer to wild salmon. So maybe it's not as far off base as it struck me. Maybe I'm just getting crappy grocery store salmon.
Great.
That's where my head is at going into this. And so I had asked Chef Adam to make me the wild type salmon like he serves it at his restaurant to guests. I wanted the full Robin experience. And that turned out to be very seasoned.
We serve it with truffle butter, truffles that we preserve, truffle salt that we make.
And I have to say, truffle's not my favorite flavor. I find it really overwhelming in a dish. It's hard not to think that the truffle is like masking something in the eating experience.
Could you taste the salmon part of the salmon?
Yes. So I recorded myself, obviously, and I tried it next to the regular salmon to compare. All right, I'm going to try the wild type. Let's see. I mean, it has the texture of salmon, for sure. But yeah, texture-wise, I would say it's pretty spot on to what I've had before in a traditional salmon. So for what I could taste from the fish, it was mild. It wasn't too fishy. Traditional salmon. Okay, so the traditional salmon, almost more, and maybe this is just the other flavors that were going along with the other piece. Almost a stronger salmon flavor in the traditionally grown salmon as opposed to the wild type.
Okay, so it seems like it was fine. I mean, I don't know, is no news good news when it comes to lab-grown fish?
Yeah, I mean, it definitely wasn't a bad experience, especially considering what Chef Adam told me about the first version of wild type that he tried.
I got introduced to wild type, my guess is like 5 years ago, when they had like essentially a prototype, and their prototype, to be honest, was like very bad. Like, this was a long time ago, right? And it was like wet beef jerky. That's like the best explanation I could give.
Okay, so yay, it's not wet beef jerky now. How about Chef Adam? How did he describe the difference between the cultivated salmon and the traditional salmon?
Yeah, I was super curious about that, and this is what Chef Adam said.
I've touched like so much salmon in my life. Probably more salmon than most people, I would say. So for me, it's like very odd.
Part of its preparation, but he didn't do it with the wild, which is something the rest of the day. He serves wild type. She said tofu than a fish. Yeah, so Renee, who's the chef in Seattle, she said she's been getting a pretty positive response from people who try it. She does say that her restaurant does cater to more of like an adventurous eating crowd, so at a baseline, people are probably going to be more open to trying cultivated meat. Chef Adam in San Francisco was a little bit more conservative. He said that the interest in trying it is about half and half the people who come to his restaurant. And there's like 10 to 20% of people who are like, absolutely not. I'm not going to try that. There's no way.
Do you have a sense of what people's aversion is?
Yeah, I mean, the big thing I was curious about in reporting this story and talking to these chefs and other people in the industry, a common thread was that it can feel very biomedical. And that's a huge reason why most people in the industry really don't like to say lab-grown meat when talking to the public.
It's not great branding.
Not great branding. The more accepted terms are cultivated or cell-cultured, because those terms lean away from like the clinical nature of things.
Even cell cultured, though, does feel a little clinical.
I think you're right. And at the end of the day, it is a hurdle for people. I think it's easier to have a positive, not always the mood when they eat out. And I did ask Chef Adam if theoretically he would consider having more menu items come from cultivated fish.
So I would like to think I would say yes, I could see a future where we have whatever 10% of the menu is 3 different types of cell cultured fish. Granted, it all. So if they have positive reactions to it, then me personally, I'm happy to serve it. If for whatever reason they don't react positively, then I don't do it.
Okay, there are all states that ban lab meat, right?
Yes, right now there are restrictions on cultivated meat in 7 states. These restrictions range from you can't sell it here to you can't even experiment on it in a lab setting. I will say that the folks that I talked to didn't seem terribly worried about these bans because right now they're so limited.
Okay, so there are obviously some hurdles for mainstream consumption of cultured meat, but I know that the other big challenge is scalability. Can this scale?
Mm-hmm. So I talked to Justin Colbeck, he's co-founder and CEO of Wildtype, and he said that right now in Wildtype's fishery, which is what they call their facility, they can produce.
So at their best, we're talking about a drop in the bucket.
Yes, and it currently takes 2 to 3 weeks to make one fillet that's chef ready. And Justin says it's being priced to restaurants to be competitive with premium Pacific salmon. Chef Adam in San Francisco said it's on par with the most expensive fish that they buy, which is fatty tuna. And what I'm hearing from people in the industry side of things is that scaling up cultured meat is really going to come down to money and investment, because more money could mean, more innovation potential, helping new companies get off the ground. And actually, in general, a lot of biotech money has been rerouted to AI over the past few years. That's just been a general trend in this industry.
Okay, given all that, what is the goal here with live meat?
Yeah, I will say everybody who I talked to who's in this industry said, we know this is not going to replace traditional agriculture or traditional aquaculture. It's just not possible. There's always going to be a demand for your traditional burger or your traditional chicken breast. The goal is that cultivated meat is just going to be another option available to people. And one thing I want to point out too is that the cultivated meat industry is really global. There's industry in Asia, in Europe, in Australia. There is a whole ecosystem for this outside of the US. And another thing that I came away from my reporting on this with was an understanding that the people involved in this really care a lot about the environment. A lot of the people I talked to were super passionate about sustainable fishing. So their particular interest in Wildtype is that maybe they can alleviate some of the pressure on the seafood industry. So for them, it's worth it to take a gamble on this product that maybe still needs a little bit of work, but they feel really encouraged by what they're seeing right now. And the people I talked to are super hopeful that this is going to become more normalized and more people are going to try it. And then they'll tell their friends, hey, I tried this interesting thing. And then it'll just kind of spread by word of mouth in some ways too.
That's exactly what happened today. Now all of us know someone who's tried it. You.
Exactly. I'm happy to be your man on the inside for this one.
I appreciate it. Thank you for your fantastic reporting.
You're welcome.
Kathleen Davis, Science Friday producer and lab grown meat correspondent. Don't go away, because when we come back, we're serving up more food innovations from lab steaks to overlooked heirlooms.
We design materials that look like, feel like a steak, and then when we put cells on them, that's where the flavor comes from.
Our food science feast continues, and for the next course, of mung beans. Last week, we took Science Friday on the road to Burlington, Vermont for a special live event with our friends at Vermont Public, and the theme was the future of food. So we're bringing you that conversation with 2 scientific tastemakers who are innovating food in very different ways. Alexis Yamashita is a community organizer studying food systems at the University of Vermont, and she's growing mung beans and trying to get them onto more plates in Burlington. And Dr. Rachel Floriani is a mechanical engineer at the University of Vermont who cultivates meat in her lab. Welcome to Science Friday.
Thank you. Pleasure to be here.
Okay. Rachel, you're an engineer. How did you get into cultured meat?
Yeah, I started off in orthopedics, so designing hip implants, believe it or not. And it was 5 years ago that I had a student come into my lab and he said, you know, the things that you're looking at, trying to move away from plastic, engineering bones and muscles, you can make food. And I was like, no, you can't. That's crazy. But after 24 hours on the internet, I found out he was actually right. And so I came from orthopedics, getting people back out on the golf course and the ski hill, to now trying to create steaks that have less of a carbon footprint and taste good to everyone.
Is that the problem that you're trying to solve, sort of the carbon footprint problem?
No, actually it's a multifaceted problem and it's going to take everyone on board. Cultivated meat is just one of the solutions. The biggest thing for me coming from Vermont is trying to feed people, especially Vermonters. This is my community, and 1 in 10 children go hungry every day. So that's sort of the goal here.
What is your lab's role? I mean, what do you do?
Yeah, so we actually create the ingredient that goes into cultivated meat. So if I just took cells from your body, it would look like goop, and that's not very appetizing. And in fact, let me retract, it's not coming from your body. Okay. So we have these cells and we have this goop and we take them from farm animals using anesthesia. But in order to have something like a steak, or you go into a restaurant and it's familiar to you, it has to look like a steak. And so what we do is we design materials that look like, feel like a steak, and then when we put cells on them, that's where the flavor comes from.
Is that the scaffold?
That's right. That's right.
And is the scaffold sort of like, I don't know exactly how to picture it. Should I be thinking of it as like the bone in a T-bone steak, or is it something different?
Yeah, that's a really good question. So it looks like a sponge. And if I were to ask you to close your eyes and touch this, you may have trouble like differentiating this from a steak. And so the idea is that really it has the same properties. That's where the mechanical engineering comes in. But it's colorless and tasteless, and then it's up to nature and biology to actually turn this into something that we recognize.
Once you make the scaffold, what happens to it?
Yeah, so the scaffold has to be cleaned and pasteurized, just like if we were to create any other food product. And then we put it into a soup pot, if you can imagine something like that. And in that soup pot is maybe some chicken broth. And that chicken broth has all the food and the nutrients, and maybe we add a little fish tank bubbler to it, okay? So the cells are getting the oxygen, they're getting all the food they need, and what they do is they find that little sponge, and that becomes their little home. And so they stick to that sponge, they crawl inside the sponge, so that eventually when you cut that scaffold open, it oozes, it
It's not that
maybe
appetizing
tastes
when you put it
good.
that way.
Use your imagination.
It oozes. Alexis, you are championing the mung bean.
Yes.
Initiate us. Tell us why the mung bean?
Why the mung bean? So in Vermont, we have many communities where mung beans are at the heart of their food cultures. And most of the mung beans that we have present in the United States are actually produced internationally and imported in. So that is really at the heart of my work, is to really cultivate a local scene where we have mung bean seeds, integrated into our gardens, our farming, and really to be able to serve more broader communities in food access with their culturally relevant foods.
Where are they native to?
So mung beans are native and grow in many different areas, predominantly in East Asia, Southeast Asia, but also Africa as well.
Do they grow well here?
Yes, they actually grow really well here. And right now, my work, we have trialed over 50 different varieties of mung beans here locally for the past 2 years. And many of those varieties are very happy in Vermont.
Are they like heirloom mung beans? Like, should I think of them that way? Like what I would find at the farmer's market with tomatoes? Or what's the difference between them?
Yeah, so they are, many of the varieties are what people call heirloom. And heirloom, there is not a unified definition of what that means. It just means old variety that's been around for many years. Many of the varieties we're working with are open pollinated. So what that means is that farmers and gardeners can actually save seed from these varieties and grow their own mung beans. And as we know, the more that farmers and gardeners are engaging with the seeds, growing them locally, they're going to adapt and be stronger to our region.
I know that you're both focused on protein. How does that fit into sort of the grand scheme of the problems you're trying to solve?
Yeah, one of the problems I guess that we're trying to solve is multifold, but the increase in the world's population. But in terms of the protein, a lot of it culture. There's a lot of data out there saying that it would be easy if everyone just became vegetarian. That's not gonna happen, right? Like, so in order to get around that, we are just trying to make it familiar. It's almost like this healthy indulgence, and the focus on protein. We have now protein infused like cheese puffs or something. It really is like how you receive the protein, and you don't necessarily need it for meat, but that's what people want.
What about you, Alexis?
So one fun fact, the common Beyond Burgers that we see, just eggs on the shelf, one of the main plant-based proteins in those products is actually mung bean. So and I know there are other products out there too that are in line with that. So there is